Our next speaker is Dr. Parth Patwari of ProteoThera. He received his Bachelor's of Science and MD at Northwestern University, followed by an SCD degree in electrical engineering at MIT. He was a research fellow at Brigham and Women's Hospital and an instructor at Harvard Medical School for 8 years. He's the founder and CSO of ProteoThera, and today he's going to tell us about the company's matrix binding peptides for targeted delivery to tissues.
Thanks so much, Yvonne, and thank you again to the organizers for inviting me to talk about ProteoThera. And I'm very excited to present to you about our platform technology for targeted delivery of drugs. It's a platform that allows us to take a systemically acting drug and transform it into a drug that's suitable for locally acting therapy. The key to the therapy is the matrix binding peptide, and this is a short peptide that, when fused to other therapies, transforms them into locally acting drugs that can be retained in the local tissues. Our lead therapies are developed to address inflammation, especially in the joints, and this leads to additional opportunities in cancer, wound healing, and tissue regeneration. We're an early stage preclinical company. We have $5.1 million into the company at this point. $1.6 million of that is non-dilutive funding from the Massachusetts Life Sciences Center, Partners Innovation Fund, and Ipsen Pharmaceuticals. So we would like to thank those organizations for getting us started. And we have now our platform technology, which we'd like to develop through collaborations and partnerships. So we're open to opportunities, including licensing opportunities. And we're also looking for investments in our lead therapies, and we have identified significant opportunities for exit windows in the next 2 to 4 years. So conceptually, the problem that we're addressing is simple enough, which is that if you can't get a therapy locally delivered to the tissue you need it, it won't work. And this simple enough concept becomes difficult for local delivery, especially to certain tissues where there's poor blood supply. And I started off in the cartilage regeneration field, and I've seen multiple therapies which were delivered in clinical trials into the joint space, and they failed to work. And the reason is that you can make things last longer in the synovial fluid or in the blood supply, but that does not mean that they will be able to get necessarily into a tissue that has a dense extracellular matrix, that has very little blood supply, and you're relying on diffusion for that therapy to get into that tissue. So this led us to develop the matrix binding fusion therapy. The key, as I said, is a matrix binding peptide. It's 21 amino acids. And it's developed from the heparin binding domain of naturally occurring molecule, the heparin binding IGF. So it's really the body's own solution to this problem of how do you make a systemic hormone or protein local. There's 2 different versions of EGF. EGF and HBEGF, they bind to the same receptor. One is systemic and one is local. So we've adapted that for fusion to any therapy. And our optimized version of this heparin binding domain binds to multiple charged proteoglycans. So it binds to both chondroitin sulfated proteoglycans and heparin sulfated proteoglycans. So we just call it the matrix binding domain at this point, and we abbreviate that MB. So we've developed fusions with multiple proteins, and this can work with peptides as well and with small molecules. So the whole point is that by fusing with a therapeutic entity, we can reach therapeutic levels within the tissues and not just within the fluids. And the other key point is that I believe this is really the only fusion technology for making something last longer in a tissue that scales from rats to the clinic. So the early work that we did with the matrix binding domain was with fusion to the IGF-1 molecule. IGF-1, a growth factor that, as I mentioned, as I alluded to, has been in the clinic, was given for intra-articular injection, so local delivery for osteoarthritis. It failed to work, and the reason is that it just does not last long enough in the tissue. In fact, it doesn't get in. And this is a Western blot showing the results of what happens when you inject either the unmodified IGF-1 or the MB-modified IGF-1 into the knee joint of a rat. And you can see that on the left-hand side here, the IGF-1 is completely undetectable inside the tissue. This is at 2 days, whereas we see robust detection of the modified version of IGF-1. It's detectable about to a week in a rat. And the other key point is that we can detect sustained activity over that same time period. So you can imagine that just making something bind to extracellular matrix doesn't necessarily mean that it's going to stay active. So it has to be able to reach the cell as well. So this is important data that if we do a similar experiment where we inject IGF-1 or matrix binding IGF-1 into the knee joint of a rat and we look to see how much new matrix is being made, we can see significant increase with injection of the modified version, the MB-IGF-1. The IGF-1 itself is not doing anything because it's just not there. So we're able to keep it there longer in a rat. We can show that this, in a proof of concept study, we can actually show that in an osteoarthritis model, this modified version of IGF-1 is active, whereas the unmodified IGF-1 is not active when given weekly in this model. And we can do this with multiple different therapeutic cargoes. So here's some preclinical data as well with our lead compound, which is the MB-IL-1RA. So IL-1RA is commercially available as anakinra. It's used to treat rheumatoid arthritis. But it's also been proposed as a therapy and been in clinical trials for local delivery, and that hasn't worked because of its short half-life. So we made a modified version, and the key point is that we're able to make a fusion protein that retains activity. There have been other attempts to make fusion proteins with IL-1RA that have led to loss of activity, so we're very pleased to see that the matrix binding domain is relatively unstructured and relatively easy to fuse to other therapies. So it has equivalent potency, and we've done preclinical work in rats and dogs, and we can show that it retains the safety profile of anakinra. There were no clinical signs after intra-articular injection in dogs at the maximum feasible dose. The other important point I alluded to before is that the scaling actually improves when we go to larger animals. And you can think of this as there's simply a larger reservoir for the matrix binding domain to bind to. So whereas in the rat, we can see a terminal half-life of about 6 hours, here in the dog, we see a terminal half-life of 3.5 days. So the technology is safe, it can be fused to other therapies without changing their activity, and it's allowed for extended retention in the tissues in rats and in larger animals. So, as I mentioned, the MB-IL-1RA is our lead therapy, and we see significant opportunities for a broad platform with this drug. IL-1 is known to drive inflammation in multiple disease states, and in particular, it's a classic cytokine for acute inflammation, but we also know now that it's the classic cytokine for inflammasome-driven inflammation. So we see this in multiple states of tissue injury, including musculoskeletal injury in gout and osteoarthritis. We see this in certain kinds of cancers that rely on an inflammatory environment, in particular adenocarcinomas. And we see this in spinal cord injury and brain injury, where we've shown, again, differential and better retention in these tissues than with unmodified proteins, and in wound healing. So there's a broad opportunity here to expand a bit on the cancer opportunity. We're very interested in this because there is published work already with IL-1 array being important to the process driving pancreatic adenocarcinoma. So this is, of course, there have been many advances in cancer with the checkpoint inhibitors, but this is one of those cancers that has been resistant to treatment with checkpoint inhibitors. Part of the reason is because there is a inflammation-driven fibrosis process that leads to dense secretion of extracellular matrix surrounding the tumor, and this not only makes it difficult to get therapies in, but also creates this difficult to reach tumor cells that hides it from the immune system as well. So systemic therapies have not been successful, and the MB domain fusion can solve this problem by delivering the IL-1 array to the dense proteoglycans that surround these tumors. And we've also established that IL-1 blockade is effective in treating some of these inflammatory conditions of the articular joints. And we know that because IL-1 array itself, the approved commercial product, is in fact used off-label by rheumatologists already to treat gout as one of the classic inflammasome-driven inflammation states. So there's also acute flares in osteoarthritis, and IL-1 array has been in clinical trials to treat these as well. But it's been widely acknowledged that the short half-life of the protein has limited the ability to commercialize this. So the MB fusion technology solves these problems by allowing for extended retention inside the joint and the tissues. So our lead indications are in treatment of osteoarthritis and gout, where we've really thought a lot about de-risking the clinical and regulatory pathway. Many therapies in the osteoarthritis space have been having a hard time getting through the FDA because if you go after a structure-modifying program, it's a very, very tough slog. So we've identified these opportunities for treating acute inflammation that will be approvable based on pain outcomes. In terms of intellectual property, we have built a very broad position. We have 6 families of patents that provide broad composition of matter coverage as well as various uses and fusion with a wide variety of therapeutics. These are already issued in the US, Europe, and Japan, and we are confident in our freedom to operate. So we're actively seeking strategic partners, not only for our lead programs, but for partnership opportunities with other therapies that may need new applications for local delivery and may need new or extended intellectual property protection. So because we're a small company, I'm pleased to plug for our outsourced commercial manufacturing organization. KBI Biopharma is actually here in Boulder, and they've done an outstanding job, and they've already helped us to develop a scalable high-yield process for the MB-IL-1 array. Our management team is led by a highly experienced team led by J.P. Moreau, who was the former Chief Science Officer of Ipsen. And as I mentioned, we're looking for investment opportunities as well as additional collaboration partners through research and licensing agreements. Thank you very much.